Okay. First, I want to say welcome to our monthly Genomic Epi seminar, and I need to apologize for a little bit of false advertising. When I sent out the reminder on Monday afternoon about this seminar, I did suggest that our guest speaker was going to do an overview sort of showcase of some genomic epi visualization tools. And I had forgotten after several conversations with Rebecca that we instead asked her to do sort of an overview again about phylogenetic trees and to specifically talk about, the application of phylogenetic trees to um, bacteria versus viruses, like how it differs, and then just sort of walk us through a lot of the considerations and means of creating these trees before having maybe a spring talk about specific genomic epi visualization tools. And then was going to give her a little bit more time to get familiar with the specific tools that we asked her to speak to us about and to provide some actual like demonstrations and tutorials of how to use them. So sorry for a little bit of misrepresentation of what the topics were going to be today, but I am pleased to welcome back Rebecca Wattam. She is from or currently works at the University of Virginia Biocomplexity Institute, and she received her joint PhD in veterinary science and entomology from the University of Wisconsin-Madison in 1992. Her interests continue to include bacterial pathogenesis and comparative genomics, and as I mentioned, she is currently at UVA-BI, and she is very involved in the Bacterial and Viral Bioinformatics Research Center, or VVBRC at UVA-BI, which is a project that is funded by NIAID, and they are actively participating in our Virginia Pathogen Genomic Centers of Excellence. And so I am very happy to welcome her back as a part of our Genomic Epi Monthly seminar series. So I will turn it over to Rebecca, and then we will have time, I think, at the end for some questions and answers. So you can either drop it into the chat as we go along or hold those till the end. Thanks.
Okay, well, thank you for inviting me. And you can all see my screen, I believe. I'm going to minimize that, I think. Oops, no, that didn't do it. There we go. Okay. So I'm delighted to be here to provide you with yet another tree talk. Now, I know you guys have had a lot of these, and in fact, I have given you at least one, and I've seen you had some of them, and I particularly chose this image because I suspect that that's how many of you feel about it. More trees, it's winter, the leaves are gone. Why are we even talking about this? Well, trees are important, especially for phylogenetic trees. And they're not only important on an evolutionary timescale, which is where I do a lot of my work, but for public health, they're very important too. So today we're going to step through generating trees, the data behind them, the different algorithms you can use, and what happens once you get the tree. How do you interpret it? What kind of things should you be looking for and thinking about? Okay. Oh, gotta go on. There we go. So I guess I don't need to tell you guys how public health workers use phylogenetic trees, but I often need to tell myself that because you have more specialized interests generally than some of the stuff I do. But we all know that phylogenetic trees are important in tracking transmission pathways. We always want to identify the source of an outbreak. And often you want to monitor not only the evolution of specific variants so that you can figure out what's going to happen and maybe have public health interventions that can, if you can see a disaster coming, you need to step in front of it. And, I was just told that you guys are preparing for Marburg, not that Marburg may come or it may not come, but it's a good thing to be prepared for that and to understand what might be on its way and understanding pathogen origins, like Marburg. So these are all important parts of what a public health worker would use a phylogenetic tree for. So how are these trees built? it all starts with data. And in these days, that data is primarily sequence data. It can be nucleotide sequence or protein sequence. But once you have the sequences, you need to build an alignment. And then the alignment is fed into a tree building method. And then the tree building method spits out a file that needs to be interpreted in a visualization method. So we're going to talk about each of these today in what might be excruciating detail for you, but it's important. My feeling is it's important for you to understand it. And every time I do something like this, or give one of these things, I find I learn a lot and it helps me understand even more about the processes of stuff I use every day. So let's dig deep into data. The trees can be built on nucleotide data, amino acid data, or some combination of the two. For the nucleotides, it can be whole genome or partial genome or a number of genes or individual genes. And for the amino acids, they can be a number of protein sequences or for individual proteins. So you have all of those combinations. Now you might ask, which is using which? Well, generally with its whole genomes, it's more of a viral thing because bacterial genomes are so large, but we'll get into those particular specifics. So basically, how does the data differ between them? Whole genome, you can use bacteria for whole genome, but we'll use different gene sets for those. But viruses, some viruses, but not all, because some viruses are big. It's a lot depending upon the size of the virus and the biology of the virus as to when you use a whole genome versus a partial genome. If the viral genome is in segments, you can't call them chromosomes, but they'll have different segments. Often you will have to use a partial genome or number or specific genes. But if it's in one single segment, you can use the whole thing. For bacteria, generally because of size, you use less than a whole genome and you're looking at the number of shared genes, although you can, and it's when you have a bunch of bacteria that are very, very closely related, you can do a whole genome sequence and look for specific SNPs, and you can do that when they're closely related, like when they're undergoing an outbreak, let's say, and you're looking to monitor changes within there, because there'll be very few changes and the data won't be so big. But, in general, you scale down on that. Now, what's common in bacteria is to use a smaller set of genes to examine things. And one of the techniques people use a lot is multiple locus sequence typing. I don't like it very much myself, but a lot of people use it, and that's a set of 7 to 20 genes that people generally use to examine different bacterial groups. And of course, individual genes are often used in epidemiological analyses of viruses. They can also be used in bacteria, especially when you're looking at antimicrobial resistance and looking for changes in that. So some whole genome sequences with the viruses. These are specific viruses here in the panel on the left that they use whole genome sequences for. And everybody knows, like SARS-CoV-2, we saw so many trees of that charting the variant tracking and how it was changing. And I did not want to include another SARS tree because we've all seen so many of them. So I use this one from hepatitis C. And hepatitis C is in a single a single segment. And this is showing that the 7 genotypes of hepatitis C, and each of these are coming from a different geographic region, and they each have different pressures. And so this is an instance where you can use a whole genome sequence for an organism like this. Now in bacteria, okay, the figure's misleading. You can use whole genome sequences in bacteria like you can with, if you're looking at an E. coli outbreak and everything's really close and you want to monitor the changes in some virulence gene, you can do that, or you're looking for specific changes. But mainly for whole genome sequences, they're used for things like endosymbionts, which are have very small genomes, but those aren't, while they are of great interest to me, they are not very interesting, or they're not at all interesting for public health workers. This happens to be a Staph aureus genome, and it's not specifically whole genome, but it's based on 1,469 genes that were seen across 135 patients in these hospitals that had either MRSA or the other one, MSSA resistant. So you can get close to that. This is a common thing we do with bacteria is we take a whole bunch of genes and then concatenate them together, do the sequences and can concatenate them together and build the tree with that. So it's close to whole genome. It's the genes that are shared amongst all of them. And you're looking for similarities and differences. That's important in all of the alignment things. You've got to have some things that are similar and different to build a tree. So if you're using partial or whole genomes with bacteria, I talked about MLST, which is multiple locuses, which is 7 to 20 genes, and the whole genome sequence, which would actually, you would use the whole genome sequence to generate the assembly and the annotation, but then you would use most of those genes for the tree. Now, like I said, MLST is 7 to 20 genes. I'm not a fan of it because if I have the whole genome, why would I just want to rely on 7 to 20 genes? But bacteriologists love those things and continue to use them. They were important in a time before sequencing became so cheap, when you would you would be doing PCR and you would use a certain number of genes because it was, and sequencing was laborious. When I first started my career, I did that kind of sequencing in the lab. We would use P32. We would put things in a sequencing reaction and then run them on these long gels and put them on a light table and read and write it down manually what the sequence was. So at that time, you couldn't really do the whole genome, especially with a larger organism. It was just too labor intensive. So MLST was good for that time. Whole genome sequencing now is much better, in my opinion, but it doesn't matter. The bacteriologists love it, and so they keep including it. And on the left-hand panel, you can see that sometimes MLST and the core genome SNPs, they have really good correlation and they look pretty similar. And on the right, you can see a different one for a different organism. And sometimes things are all whacked out and they're not so good correlation. In my opinion, I would always go with the core genome SNPs as opposed to the MLST, but I can't force people to come to my way of thinking. So when you get into viruses, you can use, we already talked about whole genomes. You can use segments, specific regions, like a region within a segment, and then individual genes. And this is very organism specific. So pay attention to the individual, and you may be confused because when you look at influenza, in the first one, segments, it says hemagglutinin and neuraminidase genes. Those are complete segments on the influenza A virus. So those are both genes and segments. For regions, you have hepatitis C, which will often use a specific region that might combine multiple genes. And for individual genes, It really depends on the organism that you're looking at, which gene you use to examine that. So one thing you have to keep in mind is that different genes can show different evolutionary patterns. When one of these organisms is replicating itself, they are often accumulating mutations. So when we're talking about the different genes, what's happening is when they replicate themselves, the an individual nucleotide might change. And if it's not a change that results in a drastic change in the protein that keeps it from doing something important, sometimes that's not corrected, especially with things like RNA viruses. Let me have a sip of water here. So you can see that these are from the same organisms. Influenza A isolates. They're from the same ones and the same colors to denote them, but they're looking at different genes. And on the left-hand side is the hemagglutinin, and on the right is the neuraminidase. And you can see that the colors, those pop out at you. So there are some different patterns there within the same organism. So you need to pay attention to that and to understand it. If you were doing a broad scale examination, you wouldn't want to use HAM for one organism and NA for another because, well, first of all, the alignment would be terrible, but the things wouldn't match up very well. So you have to pay attention and think about what it's telling you. And if you're doing it yourself, I would recommend being consistent as you go along. So that's enough about data. Once you have the data, once you've paid for the sequencing and you have all of that stuff, then we have to generate a multiple sequence alignment. And there are a lot of algorithms to do that. I've just listed a few here. I've used Clustal, I've used Muscle, I've used MAFT. They're fine. All of them are fine. Alignment protocols I find somewhat boring. People I know like tea coffee for complicated stuff. I haven't used that, but there are a lot of pipelines out there. And what the pipelines are doing are arranging things so that a tree building pipeline can look at it. Now, there are another number of methods for tree building, and please don't ask me to go into the math underneath all of these because I look at the papers and it's beyond me. But I've given you the three principal methods or the three principal ideologies underneath these methods are that define the different methods. There's maximum likelihood, Bayesian, and neighbor joining. So maximum likelihood has RAxML, IQ tree. I believe you guys use a lot of IQ tree and phyloML. I use RAxML a lot. I like maximum likelihood. I use it for most everything that I examine and that I study. And it does really well with large data sets that are complex. So I like that. Now, a lot of people like Bayesian inference, and those are primarily, well, there are other ones too, but Mr. Bayes and Beast. Beast is becoming really popular with virologists. They like that a lot. And what Beast does, it is pretty amazing, is it can put a time estimate. It's pretty good at these time estimates that they put on the branching methods. So I know the viral phylogeny is like that. It's slower than the maximum likelihood methods. So for something larger like a bacteria, it will be a lot slower. And then there's neighbor joining, which is super fast. And we used to use neighbor joining all the time and use Phylip, and I have Mega there. I've been told by people who who develop these pipelines. In fact, I was told yesterday, and in fact, these are his words. Neighbor joining is statistically naive. Well, obviously he doesn't like it, but it's supposedly less accurate than maximum likelihood and Bayesian. So if you twisted my arm and asked me for recommended methods, I would say if you have really large data sets, you want to use maximum likelihood. I like Raxamil. I know other people like IQ tree, but speed, efficiency, they're great. That being said, they can take a long time too. So, if meaning that it's fast, and the tree pipeline, depending upon the size, takes 5 days to run, it gives you an idea of how much slower the Bayesian trees would be. So if you wanted a time-scaled phylogeny to determine, how this thing is evolving, beasts, people talk about that a lot. They really like beasts, and it is popular for that time-scaled aspect and timing the divergence. And if you just want to do something really quick, do neighbor joining, something like mega. But my feeling is then you'd have to do it. I don't see that many neighbor joining trees in publications anymore, although I recognize that you guys aren't as interested in publications as the research biologists. But my feeling is I usually default to one of the other two. Actually, I always default to maximum likelihood. But for viruses, I might go Bayesian. Well, I would do Bayesian. Oh, I don't want to forget this one because this is really gaining in popularity, KSNP4. So we talked, briefly about single nucleotide polymorphisms, which is what all of these things are based on at some different level. If it's a nucleotide sequence, you're looking for the individual changes in the nucleotide. And if it happens in one, like an A changes to a T or a C or a G, that is a single nucleotide polymorphism at that place. If that doesn't change the amino acid when the protein is generated or the peptide is generated, then it's a synonymous change, meaning it doesn't have any cost to the protein. If it changes the amino acid, then it's a non-synonymous change. So all of these things are looking at these specific changes. And this particular pipeline is really coming on strong, and people love it because instead of taking the read files and looking at all the SNPs across it and comparing it to a reference, which can have its own complications. This is the thing we do for SARS-CoV-2 analysis, is we have that first reference, which is the Wuhan reference, and we compare everything to it, and we look at the changes that occur on that. And that can be misleading because the virus, we'll talk about in a little bit, viruses can change information. So you're missing that if you're using a reference. So KSNP4 doesn't use a reference, and instead it lines things up via k-mers. Excuse me. A k-mer is a subsection, like it could be a seven nucleotides, and then it would be a 7-mer or a 20-mer or a 100-mer. And KSNP4 is looking at it that way so that it's much faster than doing the whole thing. And it's been very helpful for looking at outbreak tracking, especially in Salmonella and Mycobacterium tuberculosis. People really like this thing. I haven't used it yet, but the bioinformaticians on our project have been looking at it, and they give it 2 thumbs up. So once you've run the pipeline, you still don't see the tree. You have to have another piece of software to do that. Now, I've just listed a few here. I know you guys like iTool, and I know you have a license for it. I used to really love iTool before it went behind a paywall, so I stopped using it at that point. What I use right now are Fig Tree, which is totally free, and Archaeopteryx, which is Java-based. I mean, if I had to download it myself and use it, I might not like it as much, but it's available in my resource for free, BVBRC, so you can use that there. So if we do this thing in the spring, we'll go more deeply into depth care, but this is yet something else that you need to generate a tree. So how does it all work? I mean, it seems like magic. At least it does to me. When I get sequencing reads, I don't even have to look at them. I submit them, and hopefully you have a pipeline that builds the alignment, passes it to the tree building algorithm, and then that passes it to a viewer. And then all you have to do is look at the tree. But it is a lot more complicated than that. So let's start with what you get back from the sequencing facility. There are two different file formats that come back. When this one is FASTQ files. And the first time I opened one of these things and looked at this thing, it was just, I couldn't believe what I was seeing. For every individual read, and there can be thousands or millions of them within one of these files. Each one of them has this particular format, which is 4 lines. The first line, which starts with that @ sign, has some sequence identifier and maybe an optional description on it. The second line, I hope I don't advance it. See that there? That's the actual sequence there that particular read has. And this is probably a Lumina sequence because it's pretty short. And then the third line always starts with this plus sign, and you can put, and you wouldn't be doing it, the sequencing center might put different information in there. I've never actually seen any other information in a plus sign. And then this nonsense, the fourth one, is actually giving you, these are different ASCII characters that are stand for a different level of quality, and each one of these corresponds to this. So as long as this will be equal length, and the E's mean, oh, this is really good, and the A's mean, oh, I'm not sure. The quality score for this particular read is a little bit less. When I used to, after I stopped sequencing gels myself, but there was a time when you would get back stuff from the sequencing center, you would get it, often it would give you a handout, or, you know, something that you would, a file that you'd open on your computer, and it would show you areas that it was unsure of the sequence in this. Like if I was reading a gel, it would show me places where I couldn't distinguish between the C and the T at that particular point. So this is telling you that kind of stuff. And just think about that. Thousands or millions of these. I mean, there are some, I don't want to say nerds, that go through those and think about those things, but it's just amazing. If you've been like me and from the beginning of your career doing this by hand and then having this with all this information, we really are amazing animals, and science has really brought us really far to be able to do this stuff. So here's the other one, the other format, which is a lot easier to explain and talk about. These are sequencing reads that are FASTA files, and the FASTA text format is much easier. It always starts with the, is that greater than or less than, whatever, that little sign. It has to have that on the first line, followed by whatever identifier you want after that. And the second line starts with the sequence. Now those sequences can be really long. So, whoops, let's see. This tells me that it's a read because it says forward, and this could be Illumina. It's a little, I think it's a little small for PacBio or Oxford Nanopore, but you could, I mean, this, everybody should understand this, that, you know, the CTGA, it's all there. The same format is used for a config file, which is what you have when you have an assembly, when it takes all of these thousands or millions of read files, brings them together and says, this is how I think these look in the chromosome or in whatever segment it is. If it was a config file, it wouldn't have this forward strand here. It would have more information, and the config files also have things that will tell you how well this has been covered by the read. So it'll give you an estimate of how much faith you should have in this particular segment of the genome, if it's very sparsely covered by reads or if it's really deep. And you will also, this is the same format for a protein sequence file, although that doesn't have any confidence associations with it at all, and it would have protein sequence as opposed to nucleotide sequence. So if that wasn't boring enough, the next thing we have to do is take those things and you feed it to an alignment software program, and it generates an alignment. Now you can see it as a text format, which is what the computers like to read, to see it this way, or, you know, you can see it in color, which is what the human eye likes to see, so that you can look at variation and change. And these are the same things, but seen the same sequence, the same alignment, seen either in text or color variant. And so then once you have that alignment, you feed it to the tree software, the tree algorithm, the tree pipeline, and that generates something that is called a Newick file. So what is that? Well, a Newick file is something that denotes the structure of the tree. And there are some key elements in the Newick file. This is pretty much what a Newick file looks like. And what you pay attention to are the parentheses, which show the different associations between these organisms. So the parentheses are looking at how either the common ancestors or the branching points are. And another thing you pay attention to are the commas. The commas are separating the organisms, which are established by the parentheses. Then you pay attention to the colons. The colon directly precedes a number, which is a branch length. So these guys are grouped together, or these two are separate, you know, they're two separate organisms, and yet they're grouped together with this branch length. See, the comma is separating this guy from this guy. And here we have a colon with this branch length and a colon with this branch length, and those two things are separated by this branch length, and then it all ends nice and neatly with this beautiful little semicolon at the end. Very simple, right? Anybody should be able to read this once you see it. Well, here's one of mine. Look at that thing. I mean, yikes, look at it. It's so complicated. There's some things I see here. Here's the semicolon. Okay, that means it ends. Oh, here's a colon. So this is a branch length, a parentheses. So I'd have to figure out where the parentheses was that matched that. I might have to go all the way back here. Here's another colon. Here's another branch length. Here's another colon. Here's another branch length. Here's another colon. Here's another branch length. This I know is a genome identifier. So I don't know about you. Can you figure out what this tree looks at looking at this? No, of course you can. This is supposed to be read by a computer, not by the human eye. So you have to feed this thing into a visualizer, and it will spit out something like this that we all can read. So those viewers, nobody would ever be able to read that. But if you put it in a viewer, it looks, you can see it, you can see the patterns. I made this myself. This is influenza A viruses from Virginia over different years, so that you can look at the differences between them if you wanted to. I mean, it's not that important, nothing that stands out to anybody that they get all excited about. But the tree viewer is something we should all get excited about, because that's the only way we can see anything from those mysterious Newick files. So we've stepped through all of that stuff. And you think you've got it all down, but then there are these other considerations, mostly not around the software, because you can build a viral tree with RAxML. You could build a bacterial tree with RAxML. You could, any alignment, any of those alignment algorithms, you can do viruses, you can do bacteria data, it doesn't matter. But the other considerations are, what type of data are you using? So one thing that's important in evolution and in public health is estimating the time. And you either are looking at a molecular clock, or you're examining a more rapid spread. And so when you're looking at a molecular clock, you're using evolutionary biology, and you're trying to determine when these things are separating and becoming different. And generally, when you do that, you're looking at genes or regions of the genome that are not evolving as quickly, that are on a more neutral time frame. And what do I mean by that? When any cell, it doesn't matter if it's virus or bacteria or fungal or even us, all of our cells, each time they replicate themselves, they make mistakes. And the polymerase comes through and tries to catch those, because some of those mistakes could be really bad and make the cell die. Like if it knocked out, if it changed the shape of an important enzyme that cell needs to, I don't know, digest something that's important to them, and they can't do it anymore, it's dead, and it doesn't get to leave daughter cells. So it's all very sad from the cell perspective. But some of those that aren't killers, they accumulate, all of them. Every single organism, it will accumulate mutations over time. And if an organism becomes separated by a mountain range or something, remember your basic biology, they'll have different mutations. So you use these neutral sites, the sites that aren't under strong pressure, to estimate the time of this. Now, you may think, oh, that's just for things that evolutionary biologists use. This wouldn't be of interest to public health, but think of the Zika virus, and when that came out, and they were using this kind of analysis to try to find when the change occurred between the African and Asian strains and the ones we were seeing in South America that had those devastating consequences, although they were also looking at those specific consequences to try to identify what was happening to cause that terrible morbidity in the babies. So those are the neutral things. Some genes are evolving more quickly. There are evolutionary pressures like the host immune response that can put these things, these organisms, it doesn't matter if you're a bacteria or a virus or a bird or an insect, you want to survive. So they're all responding to pressure so that they can survive. And some of these genes, the genes that are being directly exposed to, like the host immune system, if you're a bacteria or a virus, that's an extreme evolutionary pressure, and those things are often mutating more rapidly than, say, some genes or some intergenic region somewhere else in the genome. And then another thing you have to consider is that some sections of the genome are more fluid. Now, bacteria are not per se sexual organisms, but they do exchange genetic material through different processes. And when the change comes, that when a piece of a chromosome comes in that has different genes on it, that's called horizontal transfer. It didn't inherit it from its mother cell, it got it from somebody else. And this is the way that bacteria often transfer around antimicrobial resistance genes or virulence factors. So these things you need to take into account for what you're choosing to generate a tree with. And so you can have neutral or rapid evolution. Neutral evolution is when you're using those neutral areas of the genome. The mutations are accumulating randomly over time. I consider it slowly, but the rate varies in different organisms. And the branch lengths in those trees often you can use it to represent time. So you can tell how many SNPs is accumulated, and some people will fit a molecular clock onto that and be able to say this happened 10,000 years ago or whatever. Rapid evolution is when you have those strong changes and they're getting a lot of mutations that are advantageous to them, that are helping them defeat the host immune response, or, these bacteria and viruses, the bacteria are under attack. Not only, it's not like I'm a human host, so I don't care if they're under attack or not. But if you're a bacterium, they're fighting with each other. They're competing with each other for resources. If they're in the host, the host is attacking them. Viruses are coming in and attacking these bacteria too. So there are all these competition, and the genes that are experiencing this, like maybe on their outer coat of the bacteria, those ones are often directly under the most pressure. And so if you look at those genes, you can see that they are accumulating mutations much more rapidly than the neutral one. And you can't use those to look at divergence, because they would have these really long branches, which would indicate to you, if you were using it as a molecular clock, that this was a long amount of time between the divergence there. That's not what's happening. It's that the evolutionary pressure are happening, they're changing rapidly, and it could be in a short amount of time. And so you have to think about those things when you're choosing the data that you want to look at the tree, or you're interpreting a tree that somebody has shown to you. So I know that's complicated. So let's break it down and show why it matters in something like influenza. So influenza is an interesting organism. The influenza A genome has 8 segments, and each of those segments pretty much has one protein it produces. And some of those are rapidly evolving genes, which you can see up here, and some of them would be under neutral selection. So they're more shielded. The ones that are under that are under selective pressure are things like hemagglutinin, neuraminidase, the matrix protein, everything associated with the viral coat, generally, the viral capsid. And just to point out that hemagglutinin is the thing that finds the host receptor on the cell to get in. And then it gets in, it takes over the machine work of the nucleus and has it start producing viral segments. And those things are packaged up and sent off to infect new cells. So these guys, hemagglutinin is actually a part of vaccine components for influenza. So these guys, because it's important that they hit here, and it's also important when the immune, whoops, I'm going in the wrong direction. When the immune when you have an immune response against it, it's recognizing this gene. Those things are evolving more quickly so that they can respond to these hard pressures. So if you're looking at things on a large scale, like this is overall with influenza viruses and or other orthoviruses, you want to use the neutral viruses to look at that. And here's where the influenza viruses branched off from these guys, the orthomyxoviruses at this point. So you don't want to do that with the hemagglutinin gene. You want to do something, and they used PB2. And if we go back, PB2 is this polymerase basic protein. They wanted something that was evolving neutrally. Now, if you want to see why we have to something different, you want to look at things that are more rapidly evolving. You want to look at those genes. So this is hemagglutinin, and this is neuraminidase, and these are from avian isolates, and they're looking at the same organism, different genes from the same organism, and looking at how those are changing and in different parts of the world, Europe, Asia, South Africa. So, and this will be part of a bigger story that I'm going to tell you in a little bit. But also note that the branching patterns are somewhat different on these two genes. Here's Asia settled in, here are two Asian things settled in neuraminidase, but you don't see those. Those are on a different branch here. So you have to pay attention to those things. And why do we care about what's happening in birds? We'll discuss that in a bit. But if the bird flus are getting in with the mammal flus, then you can have huge problems. So you may be thinking, gosh, I'm so sick of these trees. And look at this. This isn't even a tree. These are big blobs. What do these have to do with anything? Well, I wanted to bring it home to you what's actually that the changes that are occurring in these proteins that are causing these different branching patterns have real life implications. And you look at this and you say, real life? But pardon me, but what the hell is this? This doesn't look like real life. This is the structure of the hemagglutinin protein. Remember that hemagglutinin is that thing that the vaccines are a component of vaccines that we have every year. And they're also the point that recognizes the receptor on the host cell. So this is what it looks like. And this particular area is the part that binds to the host cell receptor. It's also the part that you start targeting to create antibodies for in a vaccine. And this little pink spot and this little green spot are the actual places where you're seeing strong evolutionary pressure. You're seeing a lot of SNPs occurring there. So you can even locate exactly where that's happening. So we've talked a lot about using the SNPs in whole genomes and partial genomes and genes, but the really important part turns out to be right in two specific parts of this protein, which is coded for by a gene. So it's even particular regions can have devastating effects on these things. So the hemagglutinin gene is considered to be an antigen because we mount an immune response against it. When it starts making changes here, that's the reason why we need a different flu shot every year or every few years, because this this organism is trying to live, if you consider viruses to be living, and it wants to continue making more of itself. And so it's making these changes here that will allow it to escape an immune response, and that's called antigenic drift. And that's very important in vaccine development, and it's also generating a lot of these different changes in this gene. So if that wasn't complicated enough, there are things that these organisms do that complicate your trees. And also they complicate the vaccines. Now we've talked a lot about, well, not a lot, but we've talked about flu in the last example. So here's our little mammalian. Well, there'll be a pig this time, but it could be anything. But generally the problem is when you have the poultry farms in Asia, because the flu patterns generally start in Asia and then migrate to the rest of the world. So you have the pork farms and the poultry farms right next to each other. I should say pig farms. And let's say that this guy, this comes from a bird, and this is the regular mammalian flu virus. This came in. That's why we worry so much about these migrating birds, because let's say we have a vaccine for this guy, so it can infect the pigs. We're not worried about it. But this thing, the bird has brought in this migrating bird, some crow or something, comes and lands in a poultry farm, and it infects the chickens with a new strain that it's not particularly good in mammals. It doesn't replicate well in mammals, but it has a different hemagglutinin gene, let's say. So both of these infect this pig. Maybe it was harder for this guy to get in, but he gets in. And when it has this situation, it's okay, because different cells within the pig can be infected by the virus. That's what the virus wants to do. And so it's in fact, now I'm infecting this cell and this guy's infecting this cell. And then, you can get different strains of virus in the same cell at the same time. And when that's happening, they're both taking over, the cell machinery is taken over and it's replicating these things and it gets mixed up. So it's called reassortment. And in that case, you can get this guy, the hemagglutinin gene, that the vaccine doesn't recognize, and the mammalian strain, which replicates really well in a mammal. And suddenly you have a new strain popping up, like H5N1, that everybody is keeping an eye on and is worried about, because it's this particular situation. There's some bird components, there's some mammal components, and it's starting to replicate well in pigs, and we're starting to also see it in other mammals, including humans. So this messes up public health because you're getting a brand new virus coming in, and it also ruins a great tree because not that that's what people care that much about, but now we have a totally different hemagglutinin gene that we were comparing, and then you have to figure that out. So viruses also do so many wacky things. Well, not wacky. They're just absolutely amazing when you dig into them. It's just astonishing the kind of stuff they do. And all of this happens, template switching or breakage occurs when you have more than one strain in the same cell. And then they can do something called template switching, where you can get part of a segment, two different parts that come together. Or you can have breakage where they actually have a restriction enzyme come in that cuts that thing and then stitches it together so you get something new. So this is generating new types of the same virus, which complicates things. Bacteria too do wacky stuff. I already talked to you about horizontal transfer, which can mess up a good gene and also create havoc for public health workers if they're bringing in a very potent antimicrobial resistant gene that they're sharing to things that don't necessarily have it. I don't understand why bacteria do that. It just seems very altruistic that you would share your genes not with somebody of your same species or genus even. You might give them to somebody of a different genus and say, oh, would you like this great, let's say you're Staphylococcus, and you have this great antimicrobial resistant gene, and you see Pseudomonas, and you say, hey, buddy, would you like this antimicrobial resistance gene? You know, I don't understand why they do it. So there has to be some benefit from them. But regardless of all of that, bacteria will do rearrangements of their genomes too, which will mess up, although these don't have public health concerns too much, they can mess up a tree. They can mess up an alignment. This is from Brucella. You can see that the abortus genomes all have this big inversion here that nobody else has. None of the other Brucella. So I know I've hit you guys with a lot of stuff, but what I want to end up with is some important things for you to remember is it's all about the data that you use. The methods don't matter that much. They're not that organism specific. The tree building pipelines, the alignment pipelines, but what matters is you have to consider the data for the organism that you use. And that plays into all the different things with the phylogenies of different time scales, the practical implications, whether it's neutral or rapid, or the overall evolutionary patterns. The data that you choose is going to help you determine that. And then you may think you have it all under control, and these organisms do this crazy stuff that not only messes up a tree and has you scratching your head, but it also creates a new strain that can have devastating outcomes in public health. So that's basically all I have. If you guys have any questions, I'm happy to answer them if you haven't, run screaming from the room with yet more phylogeny.
Thank you so much, Rebecca. Hopefully you all understand why we decided at this point in the presentation, decided to have her give this presentation first before diving into the genomic epivisualization tools. I think this was very helpful to me as an epidemiologist, and I appreciate you going through some of the boring minutia of like Newick file formats and things like that I needed to know more about. So I do have one question to kick us off, but I know it is already noon time. So if you all have to drop off, it's okay. I also have a 12 o'clock call, but I will at least see if I can get one question answered, or if you all have questions that you want to ask first, I will give you, we'll stay on the line a little while.
So is that a chat question, or because I can't?
I dropped it in here so that people could read it because I tend to just be verbose. But I will read it for you. Yeah, I will read it for you if other people don't have any burning questions they want to ask first that need to like jump off.
And hopefully not like jump off a cliff after.
No, I think you really have a knack for explaining the, what do we need to know, the so what, that I think is sometimes hard to translate to the public health environment, coming from the research perspective. So I'll ask my question first, and this has to do with influenza. So since our molecular subtyping is based primarily on that HA and NA gene segment of the virus, I wanted to understand, when you're doing multi-sequence alignments for this genomic comparison to the flu viral isolates, do we ever need to examine these other, the ones that you classified as 3 rapidly evolving gene segments? I mean, I would think maybe the PB1 and PB2, but you listed the polymerase binding protein 2 as a slower evolving one, but do we ever need to look at those in addition? And then if we do that, do we just use those segments for creating additional phylogenetic trees?
I would think for public health, and I would think for public health, because to me, especially with flu, it's there in your face every year. We know where these influenza viruses are coming from. Maybe well, we don't know some of them where the bird picked it up or whatever, but I would think maybe just the rapidly evolving genes from a public health perspective, but you can always fall back on these things. The problem is that stupid reassortment, right? So you never know. It's hard to know unless you have an organism that you are damn sure has only one strain in it. You could use the whole genome then and look at all of that and do the evolutionary path, which I always tend to default to evolutionary paths because that's what I'm thinking of, the overall evolution of it. But for public health people, they're interested in the changes in those specific genes. And so I would think that the rapidly evolving genes, and everybody is using HA and NA, it seems to me. So you could even concatenate those together in the tree. I don't know that I've seen that people do that, but influenza, I just used it as a test case here. I haven't done a deep dive on it, although I'm interested in it because it is so crazy. But you can get the sequences for them, and then when you build the alignment, you just put them on it too. So you can look at two genes at once, do a multi-gene approach, but most people look like they do one or the other. So that's what I would think for public health is what you want to do is not pay attention to PB2. That guy was looking at more of a structural change, a long-term change, which you guys generally aren't interested in. But I'm really interested in, so I have to talk about things that I really like too.
Well, I just know that polymerase is it's highly prone to error with flu, but
well, they're RNA viruses, right? So the RNA viruses are always highly prone to error. They don't have the same polymerase checkpoints that a DNA virus does. So they are always much more rapidly mutating than the DNA viruses or, the bacterial organisms or human organisms. Gosh, viruses. The RNA viruses are crazy.
Even what you just said about concatenating helps me understand more, though, because when you go on Nextstrain, right, they are using the HA and NA together, and to make their builds. So they're concatenating them. Okay.
Right. And when I do that, even with bacterial, like that thing that showed the Staph aureus genes 1,459, you just concatenate those together to make an extremely long alignment. So they're all together. And in that, you can also include protein sequences and nucleotide sequences as part of the alignment. That's what we do at BBBRC for our bacterial trees, is we take gene sequences and protein sequences from the same gene. So it's like giving extra oomph to the things that are causing changes in the protein sequence. So you're giving that a little bit more weight, and you can have 1,495 genes and 1,495 proteins, the sequences for the proteins and the genes, and you push them all together into this super long alignment, and you feed that to the tree building pipeline, and then the visualization at the end. I know, sorry guys. I nerd out on these things, and it's just what I do. I can't contain myself.
Well, we are over time, but I don't want to cut anyone off. If people do have questions or things that came up in your mind while you were listening to her, you can drop them in the chat. You can come off mute, and you can always email myself or Rebecca if it's okay to drop your email in the chat. I can also provide that.
Maybe I'll stop sharing so I can see the chat if there's anything. Oh, there's the chat. Oh, that was nice. I like what Nicholas puts in. That was good. Okay, yeah, it just about tanglegrams. That looks actually that's a beautiful, uh, visually lovely. We're primates, right? We like seeing colors and they help us distinguish patterns. And what Nicholas put in the chat is a perfect example of that. And I like that a lot. I'm gonna go down and suggest it for BBBRC that they look at bringing in a tanglegram because that's beautiful. You guys could always email me too if you have a question. Here, I'll put, oh, my,
I put it in
oh,
there for
you
you,
did it.
Rebecca.
Thank you.
Yeah, I just
Okay.
wanted to make sure it was okay. All right. Well, if you all don't have anything else that's burning right away, we will go ahead and wrap up our seminar for today. This recording will be available, and if Rebecca is okay with me sharing the slide set as well, I can provide that to all invitees for this Genomic Epi seminar. And I really am looking forward to your next presentation in the spring, Rebecca, to talk to us more about all the tools that are available via BVBRC, as well as some of the other tools that we have access to and that are available free online,
And you
open
guys
source.
like iTool. iTool is your favorite, right?
Well, I don't know favorite. It's just one that we have
You
a paid
guys,
license for now. You might want
you
to ask the
guys do R?
bioinformaticians more, but we have our. And
Okay.
then the list that we had talked about was
Yeah.
our studio trees, iTool, and then Circos and Fig Tree. And you said Fig Tree is available through your site, right?
No, Fig Tree is freeware.
Oh,
It's
that's freeware.
really simple to use. Archaeopteryx is available for our site, and that's the one where I showed the
That's the
different
one.
years. So it will color the tree with the metadata, and you can color it by host, by serotype, by year, by geographic location. You can add any kind of coloring you want to those trees. So that's why I like it. Fig Tree, I've used it before we had this other thing, and I like Fig Tree okay. I will get back, I will try to get my my experience back with that so that I can say something coherent to that.
Okay, well, I will share your slides and the links that were also in your presentations after this later on today. So, thank you so much for your time and for meeting with Alex and I so that we could tailor your presentations a little bit to our public health setting. We really appreciate your time and your effort and your participation in the Virginia PGCOE. We look forward to the spring talk, and we will talk to everyone in about a month. So, thanks so much for attending today. Have a great
Thanks for
Friday and
inviting
weekend.
me. Bye.